Great. So it gives me pleasure to introduce Tim. Dr. Tim Kidd is a microbial epidemiologist who currently works within Pathology Queensland here at the Hurston campus. His work currently focuses on validation and delivery of genomic-based diagnostic solutions within PQ microbiology discipline. Tim has worked in Brisbane. He's worked overseas as well. When I was first introduced to him, I was told that he was one of the real experts in cystic fibrosis microbiology, and particularly with Pseudomonas aeruginosa. And so it's a real honour for us to be able to hear what Tim has to say about genomic-based solutions within the diagnostic microbiology laboratory setting. Thanks, Tim.
Thanks, Jason, and thanks for the invite. I asked Jason when he first approached me about this talk, I'm like, what do you want me to talk about? And he said, whatever you think's relevant and thinks appropriate. So I thought I'd actually just give a bit of an overview about what we're kind of doing in the diagnostic space at the moment with regards to Pathology Queensland and genomics. But as Jason kind of just alluded to also, I have a long sort of research sort of history also. And so I thought I'd actually finish off by also providing some insights into how you can actually use genomics to guide the development of new therapeutics. So as many of you are probably familiar with, the standard kind of pathology setting in terms of micro is really collecting the samples and using techniques that we've used for a very long time now. From say bacteriology, we actually grow the bacteria up on plates and still use the same equipment. I had a 15-year hiatus in research, and they were still using the same bits of equipment they were when I came back 3 years ago. Obviously, we do a lot of PCR testing now directly from samples, and that really revolutionized, 20, 30 years ago, how we did things in the lab. But effectively, things haven't really changed that much. From the sort of the Pathology Queensland side of things, we have a molecular microbiology department where we tend to focus on multi-drug resistant organisms. So these are things that we culture, but we want to actually characterize the organisms in terms of their resistance determinants, and also try and get some understanding about how important they might be in the context of sort of outbreaks and whatnot. But all these methods kind of have their sort of limitations. Particularly when we encounter slow or unculturable organisms, obviously, having a culture-based sort of focus is not a great thing. With regards to some of the molecular platforms, we tend to have a lot of different PCR assays across a variety of different platforms, and that really affects our turnaround times at times. Particularly with sort of the way taxonomy has gone, particularly for bacteria in recent years, because we've got all these fancy genomic technologies, we've actually understood that what we thought was one thing is often about 20 things. And our traditional methodologies, which are phenotypic-based methodologies, lack that sort of capacity to really dig a lot deeper and really tell us what the true identity of a particular organism is. And a really good example of this is, what we thought was Klebsiella pneumoniae is actually now 7 different species. So clearly, if you're looking at an outbreak of Klebsiella pneumoniae, you want to know exactly what subspecies you're talking about. With regards to resistance genes, we have what we call a CPE PCR, which looks for carbopentamases. But for every carbapenemase, we test for 5, but with NDM, which is one of the 5, there's 25 different alleles that it could be. So it's really important for us from a treatment and also a sort of an outbreak investigation perspective to actually understand what alleles there are. And I think from a therapeutic point of view, if you look at MCR genes, so these are genes that cause polymyxin resistance. MCR1 causes basically really high level polymyxin resistance, but the MCR9 that we commonly see in our local Enterobacters doesn't confer any phenotypic resistance whatsoever. So in terms of treating a patient, it's actually quite important to know these things. A lot of our methods are labor intensive. They are what they are. They're culture-based methods. And on top of this, for a long time, we haven't really had a good way of genotyping organisms in Pathology Queensland. So, clearly there's been a lot of work that's been done in microbial genomics in the research space in recent years, and really they've demonstrated some real key advantages in terms of what we can get out of these assays. First of all, they're precise, so they can allow us to really specifically characterize a particular infectious agent, but also deliver enhanced therapies to the patients, and also assist us with outbreak investigations. Obviously, they're very sensitive. So in the case where we can't grow the organism, we can actually pull an organism out. It's kind of like magic in a sense, a lot better than what we used to be able to do. Particularly for organisms like Mycobacterium that are slow, they allow us to actually rapidly identify the organism, but also provide some information about what we can treat the patient with. And we can also personalize based on, say, genotype. So hepatitis C is a really good example in terms of looking for hepatitis C variants and also resistance markers that might be that we can see in the genomic information. Clearly, with COVID, we're used to sequencing now. We know how effective it can be and how that can really enhance our capacity to really respond to biosecurity threats. And last but not least, and as I said before, I'm going to talk a little bit about this. We can use genomics as a discovery for new therapeutics. So a few years ago, there was a Queensland genomics-based sort of project that was funded, led by Dr. Patrick Harris and Brian Ford at UQCCR and PQ. And what this idea of this research project was to really take microbial genomics and see if we could implement it in a clinical microbiology service. And this sort of really laid the groundwork for what we did. So effectively what they did was build a bioinformatics pipeline that will allow us to identify organisms, look at the resistance genes that they had, but also do some genotyping in terms of MLST and also core genome clustering, so we could look sort of for outbreaks of where particular strains were transferred between patients. So they looked at just under 3,000 different multi-drug resistant organisms. And these were sort of carbapenem resistant gram-negatives, vancomycin resistant enterococcus, and ESBL producing organisms, and also MRSA. And what this study was able to do was to actually really demonstrate that about a third of the isolates they looked with, we could actually see clustering, which meant that there's quite a bit of transmission of organisms that we had no idea what was going on. And it really highlighted the need for sort of implementing this on a routine basis so that we could implement early infection control. They also did some sort of health economics sort of analyses just to look at the cost benefits of doing this. And assuming that there was about a 7-day turnaround time to get the results out to the clinicians, they estimated that there was over 30,000 less hospital-acquired infections per annum, significantly lower deaths, but a really big cost saving to the hospital. And these are very expensive tests to do, but I think this really highlights the benefits of looking at the big picture rather than how much one particular test is going to cost me. You can actually save the healthcare service a lot of money. So on the back of all this work, what we sought out to do about 2 years ago was to actually implement this into our routine service. So this required us to obviously validate the assays. We work on the Illumina short read platform. Currently, we sequence weekly using a mini-seq, but we're also currently doing validation on a much larger NextSeq instrument. We've got NATA accreditation. We got our final accreditation a couple of weeks ago, and we're basically doing exactly what the original QGHA service was providing. So we're growing up the organisms, but doing really, really good identification. So we're absolutely certain about what organism we're dealing with, characterizing the antimicrobial resistance genes, and then doing this really sort of high level, fine sort of genotyping to look for outbreak clusters. We do also have some developmental work, which we're also sort of working on at the moment, which I'll also touch on. So this involves amplicon-based viral sequencing and also metagenomics. So we're looking to, rather than having to grow the organism, actually be able to pull the sequences out of clinical samples. We do this at the moment with a Sanger sequencing 16S based method, but this is something that moving forward we'd really like to do because it's a lot more sensitive and less sort of prone to bias through PCR. So just an example of how we've been able to utilise this. Late last year, certain South East Queensland hospital had escalation of several new cases of NDM producing Klebsiella pneumoniae. This is a very concerning sort of thing to find in a hospital, and it's the last thing you wanted to get out. So this involved two different wards. We didn't really know the precise identification genotype, and we really wanted to know the allelic information with regards to the NDM allele to know whether this was truly an outbreak or just people have started traveling again and bringing these things back into the country. Previously, we used to refer these type sort of requests away, but often it would be a 2 to 3 week turnaround time. So we decided sort of pre-NATA to actually try and get these results out as quick as possible. We managed to turn this around in just under 3 days. And there was a couple of additional cases that also arose while we were doing the sequencing, which we also looked at. It turns out that 5 of the 6 patients actually did have the same strain of organism, which actually turned out to be a different species and a different sequence type from the other patients. So you can see here, this is a really elegant way of demonstrating how we can very rapidly try and understand what's going on in a particular ward. And certainly, I think because we were able to do this so well and so quickly, we most likely averted a number of other cases. So, you know, this is really sort of therapeutics in terms of infection control, if you like. One other example of how we've also very recently used this is there's a particular gentleman that suffered a very serious push bike accident, sitting on a lung transplant list, but he's got an overwhelming infection with Pseudomonas. So we've recently just sequenced an isolate because they're going to consider doing phage therapy on him, and to be able to actually provide them with sort of the characterization of the organism will allow the people that are trying to come up with this phage cocktail to actually sort of do it in a more informed manner. So moving on to the MRL. So we have the Queensland Mycobacterial laboratory that's co-located with our lab. Clearly, genomics has been a major kind of application for mycobacterial sort of diagnostic delivery in recent years, such that the WHO has now got a catalogue of drug resistance causing mutations that they publish for every year. As I mentioned before, these organisms, particularly tuberculosis, are very, very slow to grow. So often it takes up to 6 weeks to get a primary culture, and then using the traditional sort of AST methodologies can take you another 6 weeks to actually get a result. So to be able to actually come up with an antimicrobial resistance profile using genomics from either the initial culture or ultimately, hopefully, from the original clinic sample is a way of us being able to really respond and treat patients appropriately in the shortest time possible. So certainly we've been working very closely. So what we've done is we've actually sort of developed all of the processing to generate the raw sequences is done in tandem with our bacterial isolates that are non-TB. So we're looking at having a sort of a combined service so we can maximize the results for the minimum cost. And this is really just an example just to show you what their drug resistance profiling sort of reports look like. So rather than the bacteriology lab looking for, entire genes, most often we're looking for actually just sort of resistance causing mutations. So again, sort of using this single nucleotide polymorphism sort of variants to actually predict what the resistance profile will look like. And this will allow the lab to actually send out these reports much, much earlier than they would have ever been able to do using the traditional culture-based methodologies. And then just finally, in terms of other stuff that's going on in the PQ space, so our molecular diagnostic unit is also looking at trying to validate a hepatitis C genotyping and again, drug resistance analysis sort of genomics test. They're currently looking at using a deep check assay. This is all very preliminary sort of work that they've done, but certainly the first couple of runs they've done have looked really promising. So with this particular assay, rather than taking the whole sort of genome, what they do is they actually amplify up a number of fragments that are associated with either the genotype or the 3 sort of NS genes that we know that confer these resistance mutations. And then you deep sequence those, so you generate genomic libraries like we would normally do with our other sequences, and then sequence them. So with this particular assay they're looking at, it also comes with a bioinformatics workflow that's integrated. So effectively we do the sequencing, and rather than having to develop our own pipelines, we can actually input the sequence data onto their, it's basically a web-based kind of analysis platform, and it will provide the genotyping data, but also do those sort of resistance predictions like what the TB lab is doing at the moment. So at the moment we're starting off small hepatitis C, but I think moving forward, definitely our immunology department who looks at HIV is very interested in these type assays, and probably hepatitis B and some of the other, maybe some of the herpes viruses, maybe similar sorts of things that we can apply this sort of technology to. And just finally, in terms of sort of discovery, so in my former life, I was a research academic at UQ, but also in Queen's University, Belfast. And I was very fortunate to be awarded a research grant that allowed me to do RNA-seq, looking for sort of the transcriptional profile of a particular organism in a mouse model. So RNA-seq is basically us sequencing the messenger RNA across the entire genome that's generated by either a culture. So this study was really sort of like a drug discovery type sort of analysis. So effectively what we did was we had an intranasal lung infection model in mice, and we infected these mice we let the infections evolve for about 32 to 42 hours. If we leave it too long, they actually fall over, unfortunately. So this is like 2 slides of about 5 years worth of work. But when you do an RNA-seq study, what you need is a comparator. So our comparator is basically just growing it up in the laboratory, not dissimilar to what we would do when we culture from a patient. And the idea is just to really look to see what are the important genes that are upregulated or downregulated in the context of infection. So effectively, we extract the RNA from both of these samples, deplete the ribosomal RNA because we're only interested in the messenger RNA here, and then again put it onto our Illumina sequencing platforms. So with this study, we identified just under 1,000 differentially expressed genes that were either upregulated or downregulated in the context of lung infection. We found about a 50-50 split with the up and down regulated genes. And they represented about just under 20% of all the chromosomal and plasmid open reading frames for the Klebsiella pneumoniae genome. When we look at it from a principal components type analysis, we can see that the in vitro transcriptome is hugely different from that of the mouse lung, which was a really exciting finding for us because clearly there were differences. Now, Klebsiella, like Pseudomonas and Acinetobacter, absolutely loves iron in the context of infection. In fact, it's touted to be one of the most important virulence factors for this organism. And so what we know about Klebsiella is it usually has a number, particularly the hypervirulent strains, as is this one, has a number of these siderophore, basically zinc, they're basically zinc magnets for the organism. And if you take them out, the virulence of the organism is tremendously reduced. Now, a really good finding for us, which sort of really demonstrated the quality of what we were doing, was that all of these siderophore systems in this particular strain were really highly upregulated. But what was really exciting for us and a little bit surprising is that not only it turns out is iron important, but also we see zinc, manganese, and heme, is also the uptake mechanisms in Klebsiella is also. They were also all highly upregulated, as was the iron. And in particular, the main zinc kind of uptake mechanism in this particular strain is called the ZNU operon. Now, as I said before, with regards to iron with Klebsiella, they usually have a number of these different siderophore systems. But in this particular strain, and we found this across a number of other strains, it actually has multiples of the same thing. So there's redundancy in the system. So what we've done now is we've made knockout mutants for this is just the ZNU. We have done it for some of the other hits that we got that we found in our initial studies. So basically, we're taking out either single copies or all of the copies of this ZNU A operon, and just really seeing how the strain behaves in a growth limiting zinc limiting conditions. And as you can see here, when we take out the second ZNU operon, the virulence of the capacity of the organism to grow is massively reduced. It turns out that this particular gene actually sits on a pathogenicity island right next to the most important siderophore. So we're currently doing we're putting these strains back into the initial mouse model that we worked on, in addition to some waxworm infection models. And it certainly demonstrates that the zinc chelation or even zinc siderophore type antibiotics, not dissimilar to cofidrocol, might actually be therapeutic sort of targets that we could look at for this particular organism. And that's it. I'd just like to acknowledge all the people that work at PQ here, my colleagues at UQ and Queen's University, and our funders. Thank you.
Great presentation. Thank you very much. Just want to remind the audience that we're having a Q&A session at the end of the seminar. So if you have anything to ask, just throw it there in the Q&A